Hey guys, today we're going to talk about topic 1.3, membrane structure. And when I say membrane structure, we're talking about cell membranes. Now we're going to focus mostly today on eukaryotic membrane structure, just to be clear. But remember, prokaryotic cells also have cell membranes, and viruses as well have envelopes that mimic cell membranes. They're composed of similar components. So let's get started. So 1.3a, cell membrane components. And remember, you're using these slides in this lecture to complete your 1.3 notes, which you can find on the 1.3 Understandings, Skills, and Applications page. And we're going to get started right with understanding number one. Phospholipids form bilayers in water due to amphipathic properties of phospholipid molecules. Okay, so you might be asking, well, why are we doing topic one after we did topic two? And this is exactly why. Topic two, we spent talking about all the different types of molecules that build the components of the cell. And now we're going to use that information and that knowledge to really get into the structure of the cell membrane. So if you're not quite sure, or you're feeling uncomfortable with some of the words that we're going to use today, I encourage you to look back at topic two, particularly the lipids content in the proteins content, because that's really where we're spending our time. We're talking about phospholipids that make up the cell membrane, and we're going to be talking about proteins that are embedded in the membrane. So we talked about what amphipathic means in topic two, but we'll go over it again in a second. This visualization is just to remind you what we're talking about. So here's a cell, and here's the nucleus, and then there's the membrane. And if we zoom in, you can see there's this bilayer, bi meaning two, right? We have one layer of lipids on the top with the glycerol heads pointing up, and one layer on the bottom. And together, that's the phospholipid bilayer. So if you are asked to draw a diagram, this is a good representation. Make sure you're including the glycerol heads and the fatty acid tails. And we're going to spend lots of time talking about the different membrane proteins. So the cell membrane functions to separate the cell from the rest of the environment. That's kind of a no-brainer, right? Like if a cell is a water balloon, then the cell membrane is the balloon part. And just a reminder, we are talking about a bilayer. We have those hydrophilic heads and hydrophobic tails. And it's this dichotomy, the hydrophilic head versus the hydrophobic tail, that really creates this amphipathic property. The structure of the membrane is called a bilayer because it's composed of two layers of phospholipids, a top layer and a bottom layer, and those layers are inverted. And you can see here's a circle representing the cell, and you have the glycerol heads on the outside and the glycerol heads on the inside, and the hydrophobic fatty acid tails point inwards. So the word amphipathic means they have both hydrophobic and hydrophilic properties. This is an actual molecular diagram of one of those phospholipids. We have that glycerol head, which is hydrophilic, meaning it likes water, and we have the hydrophobic tail. And the fatty acid tails, being hydrophobic, means they don't like water, hydrophobic. So when you put a drop of phospholipids in water, they automatically organize to form that bilayer. And we touched on this a little bit when we were talking about the origin of life. Like, how did we get all these cellular components together from just the molecular components, right? Like, if all cells come from other cells, well, the first living cell had to come from non-living things. And we talked about the self-organization of chemistry, that certain molecules under certain conditions will perform in predictable and regular ways. And this is one of those things. So if you put a drop of oil in water, it forms droplets. And that's because that's these molecules self-organizing so that the hydrophilic heads are pointing outwards and the hydrophobic tails are pointing inwards. And let's watch that happen. So here we have an animation that shows the self-organization of lipids, particularly phospholipids in water. You have your gray molecules, which are water, and you have your green and red and blue molecules, which together are the lipids. So here you can see they're starting to move. All the green tails are pointing inwards, away from the water. So the glycerol heads, the red and the blue, are pointing outwards. And you see those tails organizing so that they don't have to touch the gray water molecules. And after a few minutes, you can really see that organization happening. There you go. And there you have it. Some pretty keen self-organization of a phospholipid bilayer. It's pretty neat, huh? All by itself. Now, there's something else important I want to talk about while we have this diagram here, or this image. You can see that the phospholipids, while the animation was still going, they were still moving. So the phospholipid membrane, it's not solid, it's fluid. Back it up a little bit. So even when they're organized and they're in that membrane structure, you can see how they still move and they jostle a little bit. These molecules do not form a solid. Now, there are molecules in their cholesterol which help maintain some cohesion so they don't like float apart, but there's no bonding that makes this a solid entity. You want that region to be fluid. So remember, the cell membrane is a fluid. Okay, so you saw in that animation two pretty distinct regions. You had the hydrophobic interior composed of the fatty acid tails, and the hydrophilic exteriors, meaning these glycerol heads, these ping pong balls, they like water, so they don't mind touching the extracellular matrix and the cytosol on the inside, because most of that's actually water. And that's amphipathic, so we have both these hydrophobic and hydrophilic characteristics of the membrane. Here you can see, I know we looked at a ton of micrographs last subtopic, but here's one. You can clearly see this is the inside of a cell, the cytosol. Here's the outside of the cell, and here's that double line. So a lot of people lost points on their cell diagrams because they didn't demonstrate the cell membrane as having two distinct lines. You have to do that because this is a bilayer. You're really drawing, the lines that you're drawing really represent the glycerol heads. You don't really see the fatty acid tails so much. So always make sure when you are drawing a diagram of a cell, please make sure that you are using double lines to represent the cell membrane, and it'll also help you remember that it's a lipid bilayer. So the membrane surrounds the contents of the cell and separates it from the environment. And remember, without that membrane, there would be no cell. There would be nothing that differentiates the inside of the cell from the outside of the cell. And if you don't have differentiation, then you don't have a cell. Okay, we're going to move on to understanding number 2. Membrane proteins are diverse in terms of structure, position in the membrane. So structure and position in the membrane. So here we have a couple of pictures of proteins within the membrane, and they have different jobs. So lots of membranes are embedded within the proteins, or I'm sorry, backwards. Lots of proteins are embedded within the cell membrane, and they have lots of different positions. So we're going to go through a series of different functions, and we're going to go through a series of different positions. So an overview of the functions includes transporters. So think back to freshman year when we talked about cell transport. We talked about passive versus active transport. We're actually going to talk about that again in subtopic 1.4 that's coming up next. So we have these facilitation channels or ion channels where molecules move back and forth across the cell membrane. We have receptor molecules, and we'll spend some time talking about those. Receptor proteins, they receive signals in the form of different molecules, chemical signals, and transmit a signal into the cell. We have enzymes that are embedded in that membrane that do particular things. So we talked about enzymes in topic 2, right? We talked about immobilized enzymes to make lactose-free milk. Well, your cell membranes have embedded enzymes, and that's just a way to make sure that we hold on to them so that we can reuse that active site again and again. Some of our cell signaling and communication, cell to cell communication, involves both receptors and enzymes working together. And then we also have proteins in the membrane that are called anchor proteins, and these help ensure that cells stick together. Think about cells in tissue, right? Your skin cells. You don't want your skin cells like floating away from each other. They need to hold on to each other to compose a barrier. So it's important that your cells can hold on to one another, and that's what anchoring proteins do. And we'll talk about examples of those. Here we have a more complex and more realistic diagram. And I have this diagram in here which shows you the different types, but it also shows you the different positions of the membranes. So we have peripheral membrane, so this guy right here, peripheral, and that means that it's sticking out only on one surface. So peripheral means like on the surface only. Now you can have a peripheral membrane protein that's on the upper surface too. Like if I just draw one, let's see. If I drew one right here, it would be embedded in the top layer, but not the bottom layer. That would also be a peripheral protein, one that sticks out on either side, but doesn't go through the entire membrane. Then you have an integral protein, integral meaning it's integral into the membrane, so it sticks out on both sides. Sometimes you'll see these called trans membrane proteins, trans meaning they span the entire length or width of the membrane. They stick out on both sides. Here you can see we have a protein channel, and that's kind of clear because you've got this hole on the inside, and that allows molecules to pass through. So when we talk about cell transport, we're talking mostly about channel proteins. Sometimes they're called ion channels. We'll talk about all different kinds. That's next, though. You have glycoproteins, which are trans membrane proteins or integral proteins, but they have this carbohydrate chain linked to them. You also see glycolipids. Glycolipids are lipids that have, so remember, our phospholipid is a lipid, so it looks like that underneath, and then it's got this carbohydrate chain attached. Glycoproteins and glycolipids are used in cell-to-cell communication. They are typically trans, meaning they go, well, the glycoproteins are typically trans, meaning they go through the entire thing. Your glycolipids might be peripheral, meaning they only stick out on one side. And then also you can see we have highlighted in here cholesterol. And you know that that's cholesterol because it's a lipid, but it's also got those little carbohydrate rings attached. And cholesterol is really the thing that helps maintain membrane fluidity. Now that sounds kind of backwards. Cholesterol is actually the thing that holds membranes together, right? It doesn't promote fluidity and flexibility. It actually regulates and prohibits too much flexibility. That's why if you have too much cholesterol in your diet, your cell membranes actually stiffen, and that makes it really hard for them to respond, and it really inhibits the movement of materials across the membrane. So one of the reasons why you shouldn't have too much cholesterol in your diet, because you want your cell membranes to be fluid and flexible. Cholesterol prevents them from being too fluid and too flexible. Okay, so again, just to review, this slide was all about the positions. We have peripheral, which sticks out on one side, be it the top or the bottom. And then we have integral proteins or transmembrane proteins, which stick out on both sides. We have protein channels, sometimes called ion gated channels. Sometimes they're called, we just call them protein channels, or transport proteins is another thing that you'll hear them called. We talked about cholesterol, and we talked about glycoproteins and glycolipids, which are combination molecules that usually stick out on one side, and they're typically used for cell-to-cell communication. So let's talk about specific types of membrane proteins. We have hormone binding sites. These are transmembrane proteins, meaning they stick out on both sides, and they act as receptors. So they are receiving signals that are sent either by the brain or by other parts of the body or even cells nearby. And the receptors themselves are actually molecules. In this case, the signals are the hormones, and the membrane protein is the receiver. So this is a cell membrane. Here we have a hormone receptor protein. It's receiving a hormone called epinephrine. And once it receives that molecule, it's going to cause a conformational change in proteins inside and then send a signal inside the cell. The point of hormones is to send messages all over the body. So these are like long distance signals. So you can think of like when you think of puberty, right? That's like hormonal. Your body starts to change and you experience all sorts of weird and funky things like growth spurts, right? Well, in order to tell your body to grow, your cells are dumb. They don't know what to do on their own. Like they're not good at understanding what they should be doing. So the brain and different hormonal glands will send signals via hormones to cells. So human growth hormone is a hormone that is received by cells. And when your cell receives that signal on the outside, right, it's got a transmembrane protein and here's your human growth hormone. It's received. That tells the cell, hey, it's time to divide because we need to grow. So hormones are long distance signals, and it's these protein membranes, these receptor proteins. You can tell that like I haven't had enough coffee today. It's these receptor proteins that receive that hormone and then tell the cell what they have to do. This is actually a picture or a representation of an insulin receptor. So this is the insulin receptor molecule. It's a protein. This is this whole big thing, right? We have the part inside the cell, and we have part outside the cell. And its job is to receive insulin. And insulin tells your cells, hey, there's a bunch of sugar in your blood. You have high blood sugar. So, hey, cell, why don't you take up some of this glucose so you can use it for all your cellular functions? This red molecule right here, that's actually the insulin molecule. So the red molecule is the insulin molecule, the insulin, and then the big blue prong, it's got the intracellular component. Intracellular meaning inside. And you have the extracellular component. That's the outside. And that's like this whole outside part. And then here's your insulin molecule. And I have this up here so you can see. Remember, this is a phospholipid bilayer, right? So here's our glycerol heads, which are hydrophilic, meaning they like water. And then you have the interior part, which are the fatty acid tails. And these are hydrophobic. Okay. Fatty acid hydrophobic tails. Okay. So I just thought this was cool because I like seeing, like these topics tend to be esoteric and kind of conceptual because it's not like you can just look at molecules and see what's happening. So I like to use as detailed images as I can so that you really understand what's happening. So once that insulin molecule binds to this receptor, then it sends a signal inside the cell telling the cell, hey, you should take up some glucose. You can think of it as like receiving a phone call. Neurotransmitters are also proteins, transmembrane proteins that receive signals. These are usually like shorter distance, like from one cell to another, like one nerve cell talking to another nerve cell. And we'll spend a bunch of time talking about that your senior year, about how that, if you take psychology, you'll learn about it too, about how action potentials are propagated across neurons and how different pesticides prevent that from happening and how it causes paralysis. Anyways, back to what we're doing. So neurotransmitters, these little plus, well, let me rephrase. Okay. Cell membrane. Here's our cell membrane, right? Here's our phospholipid bilayer. We have two types of proteins that are involved here, membrane proteins. We have our, this is a well, they're both transmembrane or integral membrane proteins. You have a neurotransmitter receiver, right? And it's going to receive a neurotransmitter. And once that neurotransmitter is received, it tells this protein channel to open up, and then ions can move in. So remember how I said earlier that both protein channels and receptor molecules, they work together sometimes? This is a good example. So you have your receiver molecule, which receives some sort of neurotransmitter, and then it tells some other protein in the membrane, like a protein channel, to open up. Ooh, I like this one. So cells don't just receive signals, right? They have to send signals too, and they also have to identify who they are. So your immune system is all about targeting cells that don't belong. So your immune system cells have to be able to recognize and identify different types of cells. So your cells have identifiers on them. Your blood type is the result of a surface protein and carbohydrate on your blood cells that identify what blood type you are. So this is actually not a human cell. This is a measles virus. And you can see the measles virus has a cell membrane, and it has these, it looks like coronavirus, but it's not. But it has these little stud things on the outside. These little stud things some of them are identifiers. These identifiers mimic the same identifiers you have on your cells, so that your immune system doesn't necessarily recognize the virus. In fact, that's one of the reasons why HIV is so good at getting inside your cells. They have the proteins on the surface have mimicked the identifiers on your cells, so that your immune system doesn't necessarily identify it as a invading pathogen or an agent. So the blue part of this is a cell membrane, right? Well, it's not really a cell membrane because it's a virus, but it mimics a cell membrane. It's a virus envelope. It's basically the same thing. We have these red and gray surface proteins. They're glycoproteins. The gray ones, I believe, are responsible for fusion, so holding on, so cell adhesion. They're the ones that actually link up to your cell, so that the virus can inject its genetic information into your cell. And the red ones, oh no, wait, I'm wrong. The gray ones are responsible for fusing to your membrane, so that actually allows the envelope to open up, and the red ones are more for like adhesion for junction. But these are there are also identifiers on here that demonstrate to your cell, like, hey, I'm cool, let me in, which is kind of messed up. Here we have another image, and this is, again, we have cell membrane, right? And we have the HIV envelope glycoprotein, which is the bottom, this thing. So the HIV envelope, that's this part. So this is a HIV virus down here, and this is the envelope of the virus. And we have, this is a transmembrane protein right here. And then this is the actual glycoprotein, the yellow and the orange. And then you've got the CD4 proteins. This is a human cell. That doesn't look like cell. Human cell. And then this is the membrane, of course, right? And then we have, these are transmembrane proteins. And here you have a glycoprotein, which again is an identifier. This is identifying, like, hey, this is what kind of human cell I am. And this HIV virus has evolved glycoproteins of its own that identify and match with these glycoproteins on human cells. And that is how HIV viruses are able to lock onto human cells, particularly the T cells. So here we have, this is an identifier glycoprotein, meaning identifying a human cell, and this is an identifier on a HIV and identifies as a HIV. But your cells don't know that. Your cells think it's another type of human cell. So it's all about the proteins. Okay, so we're going to move on from identifying and receiver molecules. And another function of enzymes in the cell membrane are immobilized enzymes. So enzymes can be embedded on either side of the membrane, so they can have either their active site on the outside or they can have it on the inside. So here you see one, and this is the inside. And remember, enzymes are catabolic or anabolic. They help either build molecules or break molecules down. And they can do that inside the cell or outside of the cell. So if their active site is inside of the cell, that means they're producing, you have your products and your reactants. Your reactants are being used to produce something inside the cell, break something down or build it up. And sometimes you have the active sites on the outside. And remember, we talked about how immobilizing your enzymes makes them more efficient, because you can use enzymes over and over again. So if they're fixed in the membrane, they're not going anywhere. You're not going to lose them. You can use them over and over and over again. And I'm going to pause for just a minute, although it's not going to seem like a pause for you guys. Okay, and I'm back. Sorry, I just got a phone call. I guess somebody at my daughter's school has COVID, but she's e-learning, so that's okay. Okay, another function of membrane proteins is cell adhesion, and we talked a little bit about this, right? So we need cells to like stick together. So adhesion proteins allow cells to form junctions with each other. And there's different kinds of junctions. There's tight junctions, which occur between animal cells. There's other kinds of junctions that form between plant cells. I want to say those are gap junctions, but they say gap junctions right here, so that's weird. It's plasmodesmata in plants, right? But these junctions allow cells to actually like hold on to one another. So if we look, you've got desmosomes, which are actually proteins that link in between. And you've got a tight junction here where you've got proteins that are embedded in both sides. You have an adherens junction. These are all examples of how proteins help hold cells together. They form like connections. It's like cells holding hands, holding on. And without that, we wouldn't be able to form tissues and organs. So that's pretty important. Then just a quick review, we have those transport proteins that we were talking about. We've got passive transport, where we're going from a high to a low concentration gradient. No energy is needed, right? These are small non-charged particles. And then we have active transport, where you're using ATP and you're going against the concentration gradient. This doesn't actually look like it's going against the concentration gradient, but it's supposed to be going from high to low, or from low to high. Either way, you're going to use energy to transport stuff. And that's those transport proteins that we talked about in the membrane. And we'll talk all about those in 1.4 as well. Okay, so for review, for understanding number 2, membrane proteins are diverse in terms of structure and position in the membrane and function, right? So protein functions. Hormone binding sites, what are they? They detect changes in the external environment sent by hormones, right? Immobilized enzymes, they have their active sites exposed on the inside or the outside of the membrane in order to catalyze reactions. You have cell adhesion, which allows cells to form tight junctions and stick together. Cell-to-cell communication, I kind of don't like that they have hormone binding sites and cell-to-cell communication as two different things, because technically hormones are cell communication, but it's more like the brain and the endocrine system communicating to your cells instead of cell-to-cell communication. I guess that's the distinguishing factor. So hormones are sent by the endocrine system, and cell-to-cell is like one cell like, hey, yo, to the cell next door. And that can be, that requires identification proteins and receptor proteins and even neurotransmitters between cells. There's passive transport, which is the transport of small non-charged particles through the membrane, sometimes via facilitated transport in protein channels. And then there's active transport, where you're using ATP to pump those particles or those molecules against the concentration gradient. Okay, let's move on to understanding number 3. Cholesterol is a component of animal cell membranes. Cholesterol in mammalian membranes reduces membrane fluidity and the permeability of some solutes. So cholesterol is a steroid, right? All steroids are lipids. So steroids, right? Anabolic, catabolic. They help build stuff up and break stuff down. Anabolic steroids help you get bigger. They're lipids. That's why if you take too many steroids, you can get it's hard to lose weight. Because remember, lipids are also used for energy storage. But cholesterol, specifically, is a lipid that's found in animal cell membranes. Now, we both produce cholesterol. We have genes that help us or that are responsible for the production of cholesterol. And we also take cholesterol in through consumption of food. So if you're eating animal stuff, you are making your own cholesterol. And in addition, you're also getting cholesterol from the food that you eat. That's why controlling your cholesterol is so hard, because you have to really police both sources of cholesterol. The cholesterol you make yourself, right? So you can take medication to help reduce gene expression that produces cholesterol or help inhibit your own production cholesterol. And you have to reduce the amount of cholesterol that you eat. So it's not just one or the other. Typically, you have to do both. But back to cholesterol in the cell membrane. They're mostly hydrophobic, and they're found in between phospholipids. So you can see this is actually a cholesterol molecule right here. They have that fatty acid tail, just like phospholipids, but they have this carbohydrate here. Now, that fatty acid tail acts as a non-liquid, more like solid, and you have this cholesterol head. Instead of having a glycerol head, which maintains fluidity, you've got this, which kind of maintains that solid structure. But it's kind of like sticking like a rod in between, and it just kind of holds everything together. You can think of the phospholipids as like rubber duckies floating on top of the water, and these guys kind of help anchor everything together. So cholesterol helps improve membrane fluidity in the rigid interior and helps control substance and diffusion through the membrane. So the saturated fatty acid tails of phospholipids behave like a solid, and the hydrophilic heads act like fluids. I just kind of said that a minute ago. And this is just an image that shows you how they're embedded in the membrane. Too much cholesterol is going to make your membrane kind of too solid. You have to make sure you have some fluidity there so that the cells are able to move and respond and be flexible. Okay, so now we're going to move on to 1.3b, which are the different membrane models. So remember, technology in terms of cell bio is relatively new with in the past few hundred years. So we had different ideas about how the cell membrane functioned before we could actually get a good idea of what it looked like. So our scanning electron microscopes and transmission electron microscopes that we really use to look at the components of the cellular membrane are pretty new. So we were stuck coming up with conceptual models of how we thought it worked before we could actually see it. That was my coffee cup, sorry. Okay. So now that we have a pretty good understanding of how the cell membrane works, that wasn't always the case. Again, here's that micrograph of a cell, and you can see that membrane represented by 2 lines. So when you're drawing your cells, make sure your membranes have 2 lines. And here we have a diagram that kind of cleans it up visually, right? So in 1935, Davson and Danieli studied the micrographs of cell membranes. So we had micrographs like this, but we didn't have any more detail. And they noted that the plasma membrane was made of 2 dark parallel lines with a light region in between. So they got that right. And from this, they developed a model of the membrane with a thick layer of proteins on either side of the phospholipids. So instead of having the membranes integral, they were all on the top, and on the bottom with like nothing in between. And like, no shame, that's kind of what it looks like, right? But then later, there were some experiments that were conducted in the 50s and 60s that provided evidence that falsified the Davson and Danieli model. And the method by which that evidence was obtained was called freeze etch micrographs or freeze etched micrographs. So what you do is you freeze a cell and then you crack it down the middle. And what that does is it splits the membrane in half, kind of like a peanut butter and jelly sandwich, like peeling apart the peanut butter and jelly sandwich so that then you have two pieces of bread, one with peanut butter and one with jelly. And what they saw is that when you pulled it apart, the inside was all bumpy. Kind of like a Biore strip when you pull it off your nose, or a blackhead strip when you pull it off and you see all those little bumps. That's exactly what they saw. So that was a good indication that the membranes were actually embedded in the center of the membrane, not just necessarily sitting on top or sitting on the bottom. Can you imagine how small of a knife you need to do that? Super small. So they noticed that globular structures were sticking out of the membrane when they peeled it apart. And they figured that those globs were made of proteins. So that kind of threw out the idea that the proteins just sat on the top and sat on the bottom, and really kind of drove home the idea that they were actually embedded in between. So as our biochemical techniques improved, we were able to extract proteins from the membrane and observe their individual structures. And these proteins they studied were globular, right? They weren't flat. You would expect if they laid only on the top and on the bottom that they would have more of like a flat kind of shape. Remember, in biology, your shape, your form is your function, right? So we tell a lot of information from your shape. If they sat on top of the membrane, you would expect them to be kind of long and flat. But they're not. They had this big round shape, which indicates that they are more likely to be embedded in the membrane instead of sitting on top. And they also had hydrophobic regions. So they had regions that didn't like to touch water. So if there are regions of a protein that don't want to touch water, they're more likely to be found in the hydrophobic region of the membrane, not sitting on top where there's water or underneath. We also did some experiments that tested the fluidity of membranes, and those experiments used fluorescent antibody tagging. So these are kind of cool. So we had a mouse cell and a human cell, and you've got membrane proteins on both of the membranes, and we tagged the different types with different fluorescent radionucleotides so that the mouse membrane proteins would fluoresce green under ultraviolet light, and the human ones would fluoresce red under ultraviolet light. So when you put the two cells together, we noticed that those membrane proteins, they mixed. And that really helped us understand the fluidity of that membrane, the fact that it kind of acts like a lava lamp, where membranes aren't fixed in one particular position, but they can move around in kind of that fluid layer. So red and green antibody tags were used to mark membrane proteins of two different cells, and then scientists fused the cells together and they waited, and after about 40 minutes, they looked under a light and they could see that all the membrane proteins had moved all around and mixed all up. Oh yeah, let me erase that. So after some time, the markers had mixed throughout the cell instead of staying in two halves, and that indicated that the tagged proteins were able to move freely around in the membrane. So again, that membrane fluidity that we talked about. Evidence from these experiments led to the formation of the Singer-Nicholson model, aka the fluid mosaic model. And this is the model that we currently use today. By the way, we're in skill 2, analysis of the falsification of the Davson-Danieli model that led to the Singer-Nicholson model. So this model describes the membrane as being made up of a fluid-like bilayer with those embedded proteins, and the proteins can move across the membrane freely. You can think of it as like a crowd in a concert where like all the different molecules are like people and they can like move around, right? It's kind of a fluid mass of molecules. Now they stay the same orientation, right? But they can move left and right throughout the crowd. Now, skill number 3 is drawing a model of the fluid mosaic membrane, right? So this is kind of like a passable diagram, but not the greatest. And let's talk about why it's not the greatest. And so, okay, let's start with the positive, right? Positive, all their lines are level with the top and bottom of the paper, and they clearly used a ruler to make those lines, and all their labels are off to one side and written neatly. Let's talk about some of the things that are not so great. So see how there's like little tails, like they made a circle quickly and there's these 2 little tails. You got to erase those. I know it sounds picky and I know it sounds annoying, but like these are the standards that IB holds you to. These aren't my personal like OCD kind of anal specifications. This is like really what IB is going to grade you on. So see how here you have like 2 lines here? That shouldn't be 2 lines. It should be one solid line. It's got to be real neat and make sure those tails are erased. Now we see the top layer, which is good. We see the bottom layer, which is good. That cholesterol, that could be better. That cholesterol should show 2 fatty acid tails and some carbohydrate rings. So you should see those little hexagons in there, right? So a proper diagram of some cholesterol would look, and there are my tails, look. I'm not practicing what I teach. Okay? And then you should have the 2 fatty acid tails. So that's what a cholesterol should really look like, right? Don't do this. That's not going to cut it. Similarly, the phospholipid tails are not that neat. You could do them a couple of different ways. Remember the diagrams of fatty acids that we talked about? You can draw them like this if you want. You can draw them, typically we draw them one straight and one bent, and then you have the glycerol head on the top. Because most phospholipids have one saturated and one unsaturated fatty acid, so one should have a bend in it. We do have some integral membranes, or integral proteins. We do have one peripheral. This peripheral is a little not so great because see this line here? This line should be like, it should be embedded on one side, right? So a peripheral protein doesn't just sit on the top. It's embedded in one of the layers, but it only sticks out on one side. But other than that, I mean, this is like, if you turn in a drawing like this, I'd probably give you half credit. So do a good job. Take your time. You've got the time to do it. Okay, so do a good job. Okay, review. By now, you should be able to answer these questions. You should be able to describe the amphipathic properties of phospholipids. You should be able to explain why phospholipids form bilayers in water. You should be able to outline the 4 functions of membrane proteins, at least 4. You should be able to describe the role of cholesterol in mammalian cell membranes. You should be able to describe the Davison-Danieli model of the membrane. You should be able to discuss the different points of evidence that falsified the Davison-Danieli model. Now, there's more than one, so make sure you have more than one. And you should be able to describe the Singer-Nicholson model. Okay? And here's diagrams of the 2 different models. All right, kids. So use this to take your notes. The lecture looks like it's running about 45 minutes. Class is about 90 minutes. So with pauses for discussion with your peers and taking notes, I think this should take you like the whole class period. All right. All right. As always, if you have questions, send me a message on Canvas or send me a remind. No Zoom today, obviously, but if you would like to have a class Zoom, we can do that soon. We are moving into cell transport. Next week, typically we do like a video project for that. It's going to be kind of fun with the Play-Doh. And yeah, if you need something, let me know. Talk to you guys soon. Miss ya. Bye.